Dr. Rauza sudah bergabung, Ivo. Bisa kita mulai? Izin mulai, Bu.
Ladies and gentlemen, may I kindly request your full attention and participation in this online guest lecture event, which is about to begin. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Greetings, everyone, and welcome to today's guest lecture. My name is Yvonne Olivia, and I am excited to be your host for this event. Alhamdulillahirabbil alamin. Praise and gratitude we say to Allah Subhanahu wa Ta'ala, who has given His grace and grace so that we can join this Zoom meeting today in Guest Lecture 2025 by Master Program in Biomedical Science, Faculty of Medicine, Andalas University. To begin with, please allow me to extend our warmest welcome to our honorable guest, Dean of the Faculty of Medicine, Andalas University, represented by Vice Dean for Academic and Student Affairs, Faculty of Medicine, Andalas University, Dr. Rauza Sukmarita, PhD. Head of the Master's Program in Biomedical Science, Faculty of Medicine, Andalas University, Professor Dr. Ilmiawati Esket, PhD. Secretary of the Master's Program in Biomedical Science, Faculty of Medicine, Andalas University, Dr. Rosa Silvia M. Clean Embryo. The moderator of the guest lecture program, Dr. Dewi Rusnita, MSc, PhD. And our special guest lecture speaker today, Assistant Professor Yanis Papadimitriou from Department of Physiology, Faculty of Science, Mahidol University, Bangkok, Thailand, and Ms. Miftahul Zanna. Ladies and gentlemen, I would like to start off by expressing my heartfelt thanks to all of you for taking the time to join us today. Once again, a warm welcome to everyone. We hope you find this session engaging and informative. Today's topic to be discussed is the role of SNPs associated with athletic success on muscle power production and health-related fitness by our special guest lecture speaker, Assistant Professor Yanis Papadimitriou, who are expert in this respective field. Professor Yanis, it is an absolute pleasure to have you with us today. Before we get started, I would like to go over some important rules for today's webinar. Please ensure that you have a stable internet connection and activate the video using a virtual background provided. Aside from when asking a question, please keep your Zoom on mute. You can also use that chat feature to ask any question on discussion session. And please keep all questions and comments respectful and relevant to the topic. Before the guest lecture, you will be asked to fill out pre-test form. The post-test will be conducted at the end of the session, so participants are asked to fill out the post-test before leaving the Zoom meeting room, as well as filling out the attendance form, and this is a requirement for you to obtain a certificate. And today's guest lecture is being recorded for future reference. Next, the following is the agenda for today's guest lecture events, and all the participants are kindly requested to follow the program with full attention and decorum. So without further ado, let's get started and let us open the event by reciting Basmalah. Bismillahirrahmanirrahim. Opening remark. An event report from the head of the Master's Program in Biomedical Science, Faculty of Medicine, Andalas University, Professor Dr. Ilmiawati Esket, PhD, is invited. Professor Dr. Ilmiawati, the floor is yours.
Thank you, Yvonne. Good morning, esteemed colleagues, students, and our distinguished guest, Professor Yanis Papadimitriou. and Ms. Miftahuzana. It is my great pleasure to welcome all of you to this special guest lecture organized by Master Program in Biomedical Science, Faculty of Medicine, Universitas Andalas. Today, we are honored to host Professor Yanis from the Department of Physiology, Faculty of Science, Mahidol University, a respected scholar in exercise physiology. First and foremost, I would like to extend my sincere gratitude to Prof. Yanis for accepting our invitation and taking the time to share his expertise with us today. Your presence here is truly valuable, and I hope this marks the beginning of a fruitful academic collaboration between our institutions. I would also like to thank the organizing committee for their hard work and dedication in making this event possible. To our students, I encourage you to actively engage with today's lecture. This is a great opportunity to gain new insights, expand your knowledge, and find inspiration for your own research. The field of biomedical science is constantly evolving, and learning from esteemed experts like Professor Yanis will undoubtedly contribute to your academic and professional growth. Once again, thank you, Prof. Yanis, for being with us today. I look forward to an enlightening session and hope that this is the start of more collaborative efforts in the future. Please join me in giving a warm welcome to Prof. Yanis.
Thank you, Professor Ilmiawati. Opening speech from Dean of the Faculty of Medicine, Andalas University, represented by Dr. Rauza Sukmarita, PhD, as Vice Dean for Academic and Student Affairs, Faculty of Medicine, Andalas University. To Dr. Rauza, the floor is yours.
Okay, thank you very much to Yvonne. And first of all, I would like to send my gratitude to Professor Ilmiawati as head of Master Program in Biomedical Science, and then the Secretary Program Study, and also to our speaker, Professor Yanis Papadimitro. So it's nice to meet you here in Zoom meeting, Prof. So I'm behalf on the Dean of Faculty of Medicine of Andalas University. Today, actually, I'm very happy because this event is very interesting, and also the topic itself also interesting. And basically, I also from my PhD program also in physiology. So maybe in next future, we can also have collaboration. Even now, I'm in Department of Biochemistry. So for the student or for all the teacher or the lecturer who participated in this event, I hope that we can get so many information from the Professor Yanis Papadimitro. And I hope also, as the head of master program said before, in the future, we hope that we can have a nice collaboration with Mahidol University. So it's a great opportunity for us, maybe not only in the this event, but maybe in the future, maybe we can also have collaboration in education, research, and also the social services. So I hope that in the future, we can have a very good relationship. Maybe today I would like to open this event officially, and I hope that this event can run well. Maybe that's all for me, and I back to the MC. Thank you very much.
Thank you, Dr. Rauza Sukmarita. Before we proceed to the main event, We will be taking a documentation session. We kindly request all participants and speakers to turn on your cameras. Okay. For the documentation session, I will count of 3. 1, 2, 3. Okay, for the next page, once again, 1, 2, 3. Okay, thank you for all your participation. Next, before we dive into the main session, we kindly ask all participants to fill out the pretest. This will help us understand your current knowledge and measure how much you gained from today's lecture. Please take a moment to access the pretest through the link provided. Well, ladies and gentlemen, while filling the pretest form, we're truly excited to dive deeper and uncover valuable insight together. We will be guided by our moderator, Dr. Dewi Rusnita, MSc, PhD, from Department of Anatomy, Faculty of Medicine, Andalas University. Our moderator today is an accomplished academic and medical professional. She holds a PhD in oncology from the University of Nottingham in UK. Please share the curriculum vitae of the moderator. Okay. Dr. Dewi Rusnita also had a master's degree in medical genetics from the University of Glasgow, UK, and medical doctor degree from Andalas University, Indonesia. She currently a lecturer at the Faculty of Medicine, Andalas University, and has previous experience as head of a primary health center and medical doctor in emergency room. Her research focuses on molecular biology, oncology, colorectal cancer, breast cancer, and biomarkers. Let's give a warm welcome to our moderator, Dr. Dewi Rusnita. We are honored to have you here with us today. Without further ado, we proceed to main event, the presentation of material by our speaker, as well as discussion session, which is guided directly by our moderator. Dr. Dewi Rusnita will help facilitate the discussion and ensure an engaging and insightful session for everyone. To Dr. Dewi Rusnita, now we hand it over to you.
Okay, thank you very much, Yvonne. Good morning, everyone. I hope you are all doing well. Welcome to today's guest lecture on the role of SNPs associated with athlete success on muscle power production and health-related fitness. This event is organized by Master Program in Biomedical Science, Faculty of Medicine, Universitas Andalas. My name is Dewi Rusnita, and I am pleased to serve as your moderator for this session. So before we begin, I would like to share a few housekeeping guidelines to ensure a smooth experience for all participants. First, please keep your microphones muted during the presentation to avoid background noise. You may type your question in the chat at any time, and we will address them during the Q&A session. And if you experience technical difficulties, try refreshing your connection or turning off your video to improve stability. This session is being recorded and will be available for later reference. With that, let us officially begin. I will start with introducing our esteemed guest speaker biography. First speaker is Prof. Yanis Dimitriou, is an assistant professor of exercise physiology at the Department of Physiology, Faculty of Science at Mahidol University. He supervises medical, biomedical, and sports sciences students. He has made major interdisciplinary contribution to several research fields, such as population genetics, exercise physiology, and applied biomechanics. Some of his research papers have been published in prestigious scientific journals with significant influence, including Nature, Scientific Reports, and several journals issued by the BMC and Frontiers publishing groups. He's doing numerous research on gene variation and its impact to athletes' performance. One of which is the latest publication about GWAS study in 2025 to uncover putative loci in predisposition to elite sprint and power performance, health, and disease. He received award from the European Athletics Innovation Award in 2015 as the winner in the open category, as well as receiving Sport Medicine Association of Greece Award 2008 and 2009 for the outstanding oral presentation. His work has been widely recognized in the field of sport physiology. The biography for the second speaker is Ms. Miftahul Zanna. Her education background, she holds Bachelor Physiotherapy from Poltekkes ERSU Dr. Rusdi Medan in 2011 to 2015. She holds Master of Biomedical Science from Universitas Sumatera Utara, and she is a physiotherapy. She holds a physiotherapy professional from Institut Kesehatan Medistra Lubuk Pakam, and from 2022 till now, she is taking Doctor of Molecular Medicine program in Faculty of Science at Mahidol University, Thailand. She worked as a staff deputy director Poltekkes Dr. Rusdi Medan Physical Therapy at Clinic Physiotherapy Dr. Rusdi Medan and as a lecturer physiotherapy of Fakulty Physiotherapy at Institut Kesehatan Medistra Lubuk Pakam. Today, Prof. Yanis and his student, Ms. Miftah, will sharing insight onto the role of SNPs associated with athletes' success on muscle power production and health-related fitness, which is a critical area of research with important implication for athletes' performance. You have 50 minutes time to presenting your topics. Without further ado, please join me in welcoming Prof. Yanis and Ms. Miftah. Prof. Yanis and Ms. Miftah, the virtual floor is yours.
Okay. Can you hear me? Hello, can you hear me?
Yes, Prof.
Yes, Prof, we can hear you clearly.
Hello, can you hear me?
Yes, we can hear you clearly, Prof.
Okay. Thank you for inviting me for this event. So the title of my presentation is The Role of SNPs Associated with Athletic Success on Muscle Power Production and Health-Related Fitness. Okay, so let's start with the nature versus nurture. Debate over whether health and physical fitness are more influenced by nature or genetics or nurture or environment is long and ongoing. So Hippocrates first acknowledged the influence of genetic predisposition and environmental factors on health and physical fitness. Modern research continues to explore the interaction between genetics and environment, aiming to understand how genetic factors influence health-related fitness and response to exercise training. Modern research initially employed the twin studies model. So with this model, initial studies investigating the influence of genetics on health-related fitness and muscle powers primarily utilize this model by analyzing the similarities and differences in certain health-related parameters, such as VO2 max and muscle torque production between monozygotic and dizygotic twins. So researchers could estimate in this way the heritability of these traits. So as we see on these two figures, the main traits that they focused initially was the maximal oxygen uptake, or VO2 max. So this is a very important health-related parameter. And on the second figure is the torque development or torque production, which is a very important parameter because torque and power declines more rapidly as we age compared to muscle strength, for example. So that means that muscle power is much more important than strength. So as we see from the first figure, the intra-pair differences between monozygotic and dizygotic twins. So it's obvious that in the first figure, the black dots are the monozygotic twins. So the intra-pair difference between those twins are smaller, like than the intra-pair difference between dizygotic twins with regards to maximal oxygen uptake. The same we can observe on the second figure, as we see in this case here, the dizygotic are with a square symbol, and the monozygotic are with a triangle. So we see the triangles are more closer to the line, which means that intra-pair differences are smaller in this type of twins that share that have almost identical DNA compared to dizygotic. So this type of research on twins concluded that heredity contributes around to from 50 to 70% of heritability in VO2 max and muscle torque generation, respectively. So that was the first methodological approach to the question. A few years later, like another study model has been introduced. So as we see here, with this model, they employed family studies. So, and as we see in this graph, all these dots, the red dots we can see here are, there have been some participants that responded to this training stimulus very well. So up here in the graph, so these dots are the participants that responded very well. And below there were also participants that they didn't have so great response. So they responded below average, so which are on the lower part of the graph. So the columns, these columns here, like a pinkish color, they are families. So an interesting observation here was like all the participants that responded very well, they were members of the same families, so which is at the upper part of the graph. And the participants that they were low responders, they were also members of the same families. So this clearly shows that response, not only the initial levels, but also the response to exercise training is highly heritable. So, but in that time, like in these studies, mainly took place in 80s and 90s, so it wasn't really the technology hadn't been evolved that much in order to be able to identify specific genetic factors that influence response to training or initial levels of performance. So, but after 90s, when the PCR was discovered and widely applied in the field of biomedical science, so scientists were able to identify specific genes that influence athletic performance. And in that time, from like about 90s that PCR was discovered till today, they have been identified about 251 DNA polymorphisms that have been associated primarily with athletic success or elite athletic status. So the majority of these studies used the case control study approach. So which means that they had a group of very successful athletes and a group of general population, and they compare the frequency of certain common genetic polymorphism between those groups. So if the frequency was higher in elite athletes compared to control, they concluded that this SNP or this polymorphism probably influences performance. So this study design gave all these 251 DNA markers that are related to these traits. But this study design has lots of limitations. So case control studies, they don't really quantify the influence of these polymorphisms on performance, but it's just able to maybe identify, but they can't quantify the amount of how much each polymorphism influenced the performance. So they can be biased. So there are many limitations. So there are two exceptions. Most of these studies follow this case control study approach. Apart from those studies, one is this Nature paper that I co-authored, and another paper that was involved elite sprinters, but it wasn't case control. It was a quantitative approach. So our main aims as a research group is to go down that line and use the more quantitative approaches, and deepen our understanding of the relative powers of common DNA variants linked to athletic success, focusing on their impact on health-related physiological characteristics such as muscle torque and power and VO2 max within general Southeast Asian population. That's at the moment, our main aim, but we also aim to identify using this quantitative approach and novel SNPs that influence these traits. So as a research group, you use innovative studying design and crossing scientific specialties is a cross-field approach. But before I go to some of the SNPs that we're going to present to you today, I just want to give you some basic knowledge, some background information. So common genetic polymorphisms, more often single nucleotide polymorphisms, are heritable nucleotide differences between individuals that occur in at least 1% of the population. So they are not rare, so they are common. We can see the figure 1 some examples, and also in the figure 2, I want to just explain to you that some of these SNPs, they have no effect. So they are outside of genes. And there are two types of SNPs. Then some of them are within the regulatory sequence of a gene. So which means they don't change the protein, but they can influence the amount of protein produced in the body because they influence, for example, transcription factors that regulate how much protein is produced. But if they're within the exon areas, then they can directly influence protein, or they can make a protein non-functional. So they would be completely deficient, the body of this protein. So scientists are identifying and studying this small genetic variation among humans as they can influence health-related fitness and explain difference in susceptibility to a wide range of chronic diseases. So some of the SNPs I'm going to present to you today. Actually, I'm going to present to you two SNPs, the ACTN3 and A SNPs that have been working for about 20 years. And the rest of the SNPs that we are going to present to the rest of the SNPs, my student are going to present to you after I finish the presentation, this presentation. So I will present to you about ACTN3 and ACE gene, and my student will present to you about the myostatin gene, the VDR gene, the NOS3, and the CKM. So she was very enthusiastic and she wanted to join me with this presentation. So I give her opportunity to be part of this event. So let's start with ACE gene. So the ACE gene initially were identified in climbers, in elite climbers. So the very elite climbers happen to have more higher frequencies of the I allele of this gene. So as we see here with red, so while in general population they were they had both, they have ID and also DD, the DD genotype. So the I allele was more frequent on this population of elite climbers. So the ACE ID polymorphism can influence the levels of ACE enzyme and thus affecting athletic performance in high altitude and cardiovascular health. So on the other hand, a few years later, this was the first SNP in 1998 that had been discovered. So in 2009, my research and also others identified that the D allele of this SNP was of this polymorphism. Actually, is not SNP; is just has like a length of about, is an insertion deletion actually. So the D allele was more frequent in elite sprinters. On the other hand, so as we see from the basic physiology, so this influences the renin-angiotensin system. So the ACE enzyme is produced by the lungs and kidney. So, and converts angiotensin 1 to angiotensin 2. Angiotensin 2 is more potent than angiotensin 1. So it can cause vasoconstriction in the body. So when you have the I allele or the II genotype, you're producing less angiotensin converting enzyme, which means less angiotensin 1 will be converted to angiotensin 2. And then if you have less angiotensin 2, then you will have more vasodilation, which means more oxygen will be delivered in your working muscle, and this can affect your performance. On the other hand, if you have less angiotensin 2, then you will have more vasoconstriction in your body, and maybe and also a higher sympathetic activity. So and this can deliver less oxygen to the muscle, to your working muscle, which means you will perform not that good in endurance activities. But maybe that would be an advantage if you have the D allele, which means you produce more angiotensin 2, so you have higher sympathetic activation. So that's an advantage over sprinting and activities that require power and more sympathetic activation. So the angiotensin converting enzyme is a key component of the renin-angiotensin system, and this polymorphism influences the enzyme synthesis in the body and affecting various healthy related outcomes. Another interesting gene that initially identified in elite athletes was the ACTN3 RX polymorphism. So this is a single nucleotide polymorphism. So as we see here, in Australia, this gene was first identified. So the power Olympians, which means very elite athletes, were more likely sprinters, and power athletes were more likely to have the R allele or the RR genotype, while the XX were completely, not even one of them was identified to have the X. On the other hand, the endurance athletes were more likely to have, with black bar here, the XX genotype. Okay, so in 2003, that was initially. In 2008, I replicate this in Greek athletes, so I also observe the same finding here, that the elite power-oriented athletes in Greece had higher frequency of the R allele or the RR genotypes, but on the other hand, I didn't find endurance athletes to have higher frequencies of the X allele or the XX genotype, in contrast to what they found in Australian studies. So, but the main thing that this gene influenced, like the power and sprinting performance. So after this, gene became very well investigated around the world. So as we see here from Australia, the first study, the Finnish, the Greek athletes, starting in US and Russia and pretty much most of the countries in the world, they wanted to investigate the influence of this gene on athletic performance using the case control study model. In Australia in 2019, so we me and my team conducted biopsies in humans, in human muscle, and actually we wanted to see the alpha-actinin protein in human muscle, which is the protein that produced from this gene. So this SNP is within an exon. So as we see here from the Western blot, the RR, they produce the alpha-actinin protein, while the XX, they are completely deficient. So we couldn't detect any amounts of protein in their muscle biopsies we took here. So that was the first study that in humans that identified that this actually, the humans that have the XX phenotype, the XX genotype, they are completely deficient, which means this gene within an exon makes this protein non-functional. An interesting finding was that we had here is like those that they didn't produce at all alpha-actinin in the muscle, they overproduced another similar protein, the alpha-actinin 2, as we see here with Western blot, there is darker this. So this means that their body, the human body, when doesn't produce this protein, compensates its absence by overproducing a similar protein trying to compensate the absence of alpha-actinin 3, but probably doesn't do this very well. So the structural role of alpha-actinins, as we see, is like they're located in the Z lines of the sarcomere. So there are structural proteins. Don't confuse them with actin. So actin is a different protein. So alpha-actinin is located in the Z lines of the sarcomere. Probably this structural difference changes also the function, but apart from this, they have also functional role because they're interacted with so many proteins in the sarcomere. So apart from the Z line, they're interacting with thin filament, intermediate filament, sarcolemma, and most importantly, with various metabolic enzymes, such as calcineurin, calcakins, and so they can also influence the metabolic properties of the muscle. So in our study design, so we recruit participants, Southeast Asians. So then those that have the inclusion criteria, they're appropriate for inclusion in the study, they're eligible. We separate them in the different genotypes, and then we do some testing using biomechanical analysis and VO2max as well. And at the end, finally, we do a genetic testing. So I will just explain to you about all these steps. So initially, we're collecting blood mostly by this method because this is less invasive and yeah, can give us quite a good quality of DNA. So after we use we're isolating the DNA using magnetic beads. So magnetic beads consist of paramagnetic particles coated with materials that they can bind DNA reversibly, allowing for the isolation of DNA for the wide variety of samples, including blood, with minimal processing without need for centrifugations. So this can give us a good quality DNA and is like a quite new technique for isolating DNA in the field. It's simple and doesn't cause damage to the DNA because we don't use centrifuge and we just use magnets to attach the DNA. So for the genetic analysis, we use a multiplex PCR, which is a technique that amplifies several DNA sequences simultaneously in a single reaction using multiple primer pairs and a temperature-mediated DNA polymerase in a thermal cycler. With this method, we can detect and amplify up to 800 different gene polymorphisms, which then can be identified using a mass array system that couples mass spectrometry with first published in Frontiers, and it's like a unique way that we can approach and collect power and torque related markers. So we use these formulas here that we developed specifically for this purpose to collect this torque and power related performance traits. So as we see here, that's what we get from the participant, from the cameras on the screen, and then we just measure the torque on the knee and hip joint primarily. So for the measurement of VO2 max, we're using a Quark CPET metabolic system made by Cosmed. So we can measure, we can do electrocardiogram during exercise and measure various variables such as blood pressure and different other variables during the VO2 max tests. So the preliminary results that have been published in this journal here, we found that the DD from the H gene, they produce more torque in their hip joint. For the ACTN3, there is a trend for higher amounts from the RR individuals, but because the sample size is still very small, we can't really have any meaningful conclusion, but we recruiting participants in order to increase the statistical power and have more meaningful results in the future. So that's here is the most of the parts of my team. And so in conclusion, genetics influence human physiology and health-related fitness. And this presentation points out some of those genes. So my student will further continue with this and present to you which genes she wants to study, actually. So the future perspectives from the case-controlled study model, we have moved in more quantitative approaches in 2024, from 2015 to 2024. So the future is like interdisciplinary. So we want to use technologies from field of biomechanics, from field of physiology, for field of medicine. So only with an interdisciplinary approach we can probably achieve our aims. So genetic testing alone is still not a reliable predictor of athletic performance. Future research may be more effective by focusing on innovative cross-field approaches such as the speed gene study and influences of these SNPs on certain health-related fitness characteristics. So we need to focus more on influence of these SNPs of certain health-related fitness characteristics rather than aiming on elite athletic performance only. So implications, the acquired knowledge will likely have important future implications in biotech industry with focus on gene therapy to combat age-related muscle power or cardiovascular fitness decline. personalized preventive medicine, and will drive advancements in exercise program design. So that's my part. My student will now join with presenting you more genes and what SNPs she really wants to do in her thesis. And yeah, so that was pretty much from my side. So she can continue, and maybe at the end we can ask me questions.
Yes, thank you for this time for me. Can I continue now? Okay.
Yes, please.
Yes. Yeah, it's a pleasure for me to be here today. Thank you for my advisor. And this time I want to discuss several variants of the SNP that have a polymorphism, that one an existing topic in this field of the muscle physiology and genetics. The gene polymorphisms and their impact on muscle function. For the specific gene polymorphisms that influence how our muscle develop and response to exercise and recover from fatigue. In this presentation, we will explore some of the key gene polymorphisms that I want to analyze for the further research in this my PhD. So as you can see here, this figure appear to depict a child with someone, or this baby actually have myostatin. It means it will make her muscle will grow so higher. And on the other hand, in contrast, a frequent sequence variant such as like K15R polymorphism is the one of myostatin gene receptor, was identified by one researchers that as a possible marker for analysis of muscle-related phenotypes and associated with might be influenced power production and muscle hypertrophy. So the one study for myostatin polymorphisms and influence on elite athlete status is showing on the graph the results. So they found arginine allele, R allele, on the sprinters was significant higher in top elite athlete for sprinter compared to the national level in the sprinter. By this finding, the myostatin location, according to the human genome reference, this relatively small gene is located on chromosome 2, specifically on chromosome Q32.2, and it between the coordinates 190.52 until 0.63 base pair. And in this myostatin include only 3 coding exon. There are 3 coding exon that have several polymorphisms. The most common polymorphisms is the 4584 A2G allele. For the one study, they found around 7% minor allele frequency in the world population in among Europeans, Africans, Americans. So the myostatin production or synthesis, myostatin polymorphisms are the one of the member from the transforming growth factor beta or TGF-beta family. Myostatin is a skeletal muscle specific that was encoded and secreted the peptide that modulates myoblasts and then can regulate the proliferation, which is encoded protein that inhibit muscle cells differentiation for the muscle growth. So by this myostatin TGF-beta family, the myostatin have a functional role function for the myostatin. It has 2 component of the myostatin function. So this figure illustrate the signaling pathway that involve muscle growth regulation. So particularly the inhibitory role of the myostatin in the muscle development and the promoting role of the IGF-1 in protein synthesis. So on the left side of this figure, as you can see, it showing the inhibition of the muscle growth. As the myostatin act as the negative regulatory of the muscle growth, it will bind to the activin type 1 and type 2B receptors and will activate the SMART signaling cascade and phosphorylated SMART2 or 3 and bind to the SMART4. And this activated will translocate it to the nucleus and inhibit the expression of the myogenesis that related gene such as the MyF5, myogenin, and MyoD. This essential for the muscle growth and differentiation. And the right side of this figure is showing a promote of the muscle growth. It's showing by the IGF-1 promote to muscle growth. It will activate the IGFR. That activation stimulate the PI3K as PI3K pathway. It will bind to the AKT signaling pathway and it will showing the AKT phosphorylated. So in this subsequently activated, it will activate the mTOR and promote the protein synthesis, and this way it can cause the and lead to the muscle growth. So the next gene is the CKM. CKM gene is the one creatine kinase reversible catalysis, the transfer of the phosphate between ATP and creatine phosphate. Several polymorphisms with the CKM gene has been associated with increasing of the CK catalytic activity. So in this part, it is showing like in oxidative phosphorylation in mitochondria. So one question for us, how CKM can restore energy? So this figure out represent the process. It starts from the creatine kinase, that creatine kinase system of the phosphocreatine, which is one of the crucial role in cellular energy transfer, particular between the mitochondria and the cytosol. In this system, it will help maintain ATP supply to the cellular function, especially in tissue and with high energy demands. So in this mitochondria, oxidative phosphorylation generate ATP, and ATP use the mitochondria creatine kinase. So this pathway can help store high energy phosphate in the form of the phosphocreatine, which can be transported to the cytosol. It means the breakdown of the ATP can accumulate, which is the recycle back to the mitochondria, and ATP can regenerate. So the one study for the CKM analysis for the CKM polymorphism, especially for CKM RS811, this is the one snap of the researchers do. They found any differences between two allele A to and G allele that comparing from the athlete and the non-athlete in Russia and Lithuania. So they found A allele is more higher than G allele. It's showing us the position of the gene CKM polymorphisms is located on chromosome 19, specifically for the 19q, 13.2, and 13.3 for the function in skeletal muscle and heart muscle function. So for the specific function role of the CKM enzymes, it's showing the role of the phosphocreatine systems in muscle contraction. An energy metabolism during exercise is showing the mitochondria process. The produce of ATP, which is used by creatine kinase to phosphorylate creatine into the phosphocreatine. So phosphocreatine can move to the from the mitochondria to the cytosol, and it will donate the phosphate group to the ADP via cytosol creatine kinase and regenerate the ATP. So by this way, creatine will recycling again, and after donating the phosphate, the creatine return to the mitochondria and to be the rephosphorylated, as we can see on the blue bits here. It shows the actin and the filament here that's showing the regulatory of the ATP process. And during the muscle contraction on exercise, ATP demands increase, so phosphocreatine rapidly regenerates the ATP to sustain activity. This system can help maintain ATP level to prevent fatigue during short and intense exercise. So the other gene is NOS gene. This is nitric oxide synthase enzyme that catalyze the production of the nitric oxide from the L-arginine. So the regulation it's come from the endothelial cells. The acetylcholine activation binds to its receptor on endothelial cells membrane and leads to an increase in intracellular calcium ion concentration. So nitric oxide synthase by the rise of calcium ion activate the nitric oxide synthase and an enzyme that convert L-arginine into the nitric oxide in endothelial cells. So nitric oxide can activate the guanylyl cyclase in the vascular smooth muscles and catalyze the GTP to convert to the cyclic GMP. So by this pathway in a lower intracellular of calcium ion level in the smooth muscle cells. So when any decreasing of the calcium ion, it will cause the muscle relaxation and leading to the vasodilation. So how about the blood regulator by the blood pressure and blood flow? The nitric oxide as a vasodilator can help to reduce vascular resistance and increase oxygen delivery. So the one study that already done for the nitric oxide polymorphisms, NOS3 gene polymorphisms, especially that start from the promoter region, the polymorphism found that NOS3 786 TC allele found that any significant differences exist in NOS3, especially for the endurance. It is the higher frequency on the TT allele. As we know, NOS3 location on chromosome 7 for the specific location on 7q35 until 36, it contains 26 exon. So it has 3 specific polymorphism in NOS region that are the on promoter region for TC allele and for the in intron region and on exon region. So the most common polymorphism occur in start from the promoter region. It is the one study that already done before. It will affect for the protein synthesis and it will produce more protein that correlate with the skeletal muscle metabolisms for power and for endurance. So nitric oxide has several protein interaction. That's many protein interaction that interact with NOS3 polymorphisms. That we need to focus for the one protein interaction is the KDR. KDR is the kinase insert domain receptor. It is the one of vasculature endothelial group factor of the receptor 2. It means in this part, we can know how the NOS3 rules for the nitric oxide signaling. In endothelial nitric oxide synthase, it will encode by the gene NOS3. It will generate nitric oxide in the vascular endothelium. Nitric oxide acts as the signaling molecule to activate soluble guanylyl cyclase. It's the one of heterodimer protein with one subunit encoded by the gene GAC1A3. So this cyclic guanosine monophosphate produced by soluble guanylyl cyclase then activate downstream signaling molecules that leading to the vasodilation, blood pressure will be lower and inhibit the platelet aggregation and other cardiometabolic effects. So this in this situation, we can conclude when someone have higher iNOS activity, it means more nitric oxide. It will increase blood flow to the muscle, and when nitric oxide activity is in lower, it means nitric oxide reduced, so it will have a limited oxygen and nutrient supply. So in low blood flow condition, it will lead to the adaptation of the fast twitch muscle fiber, especially for type 2. It caused less oxygen dependent and impact for the type 2 fiber rely more on anaerobic metabolisms. This safe might be enhanced explosive power, but reduce the endurance. So the other gene is the vitamin D receptor. Vitamin D gene receptor is the one gene that provides instruction for making protein called vitamin D receptor gene. For the more specifically, the VDR protein binds to the active form of the vitamin D. It's a calcitriol. So this interaction allow the VDR partner with another proteins that called retinoid X receptor or RXR. So its result complex then bind to the particular region of the DNA as known as vitamin D response element and regulates the activity of vitamin D response genes by the turning these genes methylation for gene on and turn off. By this complex help controls calcium and phosphate absorption, as well as other physiological process such as muscle contraction will happen by this pathway. So this is one study for VDR gene polymorphism is FOC1. It has C to T allele polymorphisms that can influence in elite status. This study found any differences between C allele to T allele. It has T allele more higher than C allele frequency in sprinter comparing to the endurance and control. So for the gene location, it's located on chromosome 12, or specifically chromosome 12q13.11. It contains 9 exons in start from promoter region, encoding region, and untranslated region. So for the most common polymorphisms for VDR gene is encoding region. It is for SNPs that has the one of the common is FOC1 gene polymorphisms. So for the functional roles, how the VDR gene polymorphisms in skeletal muscle contraction. As we know, the ATP production for the calcium ion uptake of isolated vesicle in sarcoplasmic reticulum was reduced in muscle. So calcium is the one of the second messenger that can involve in muscle contraction. That calcium must be represent here inside the muscle fiber. So in skeletal muscle, calcium ion is stored in sarcoplasmic reticulum that can depolarize by the sarcolemma. It's the one of the local current travel along the tubules and cause a voltage dependent by the dihydropyridine receptors. So it will cause the opening of the calcium channel on the surface of the sarcoplasmic reticulums. Since the sarcoplasmic release from the SR is one of the key driver of the type 2 muscle fiber of fast twitch to activate. So individual with the F allele in FOC1 polymorphisms may have a better anaerobic performance due to enhanced calcium flux. Okay, so that's all my presentation about all the gene that I'm interested to analyze in my PhD under Mr. Yanis. Okay, thank you for your kind attention.
Oh yeah, thank you so much, Prof. Yanis and Ms. Mifta, for such an insightful presentation. Both of you cover some genes related to muscle production, which is important for athletes' performance. Now we will move on to our Q&A session. We have received several questions from our audience through chat box or via Google Form. I'll read them out. But first, I would like to say that if you would like to ask a question directly for the audience, you can raise your hand, or if you are shy, you can post it in the chat box, and I will collect it and post the question to Prof. Yanis and Ms. Miftah. Is there anyone who wants to pose question directly? Nope. Okay, I'm just gonna start with the question.
Sorry, I can't hear anything.
Oh, sorry. So the first question is coming from Dr. Muhammad Reza. What contribute to muscle strength? Since muscle size is not directly related with the strength and endurance of the muscle itself, I experience some athletes with less muscle, but have a stronger muscle and more endurance in the field. How can you explain that, Prof? Yes.
Yeah, actually, I mean, from my understanding, so muscle size and muscle strength are not that related. So, and you wondering, so if they are related or not, whether or are related or not. So there is some relation. I mean, if you have like larger, more muscle mass, so you can possibly, there are more chances to be also stronger, but it's not that being having larger muscle mass is the only factor that makes you stronger. Okay, so there are people that they are not that muscular and they are very strong. So the reason is because there is other factors such as like neural activation. So the nervous system also influences and controls the muscle function. So, for example, certain people are able to activate all the motor units simultaneously of the muscle. So even they have not that big muscles, because they can use this skill to control all motor units at the same time, they can produce eventually more force than someone that have large muscles, but he's not trained to recruit all the motor units of the muscle or most of the motor units simultaneously. Okay, so it's also neural factors that influence muscle power production, not only the size. But size is also an important factor. Okay.
Okay. Maybe Ms. Miftah, you want to add some more? Hello?
It's okay.
Okay. We can proceed to the next question. This is from Dr. Dharma Yosua Sardol, SPOG. It's an obstetrician. Is it still possible to develop muscle size and strength at the age of over 40 years for women and men with diet and exercise? And if yes, what exercise options can improve this? And muscle size and strength, can it be passed on to the next generation? Could you share your thought on this, Prof. and Ms. Miftah?
I mean, at power and strength, but the decline of power is much faster, and that's why it's even muscle power is not considered to be a health-related fitness characteristic and considered to be skill-related, basically. But because it's so important for, because it declines so fast as we age, so the new guidelines for ACSM has included muscle power as an important like factor that we have to focus on. So there is lots of research like focusing on muscle power now because it declines as with age. But you can still develop them after 40, both power and strength. Yeah, so the exercises like you can do for developing them, I mean, from resistance training for muscle strength, and you can develop muscle power with more functional exercise, such as medicine balls and certain like more specific exercise for developing and maintaining muscle power. So also a lack of muscle power, research have shown that is related with increased accidents during aging. So people that have more severe types of sarcopenia and faster decline of muscle power, they are more likely to have falls and different types of accidents. That's why improving this is crucial.
Okay, speaking about the muscle injury that you mentioned recently, is there any specific SNPs that is that can prevent someone or athletes from muscle injury?
It have been studies with SNPs and like muscle injuries. ACTN3 also is because it's the R allele is producing the alpha-actinin protein, and there are some studies show that having the R variant of the alpha-actinin, the R allele of the ACTN3 gene can help you have less injuries in sport, and also with aging. Studies with ACTN3 have shown that people that have the R allele and producing the alpha-actinin protein in their muscles, they have less accidents during aging compared to individuals that they are deficient and they have the X allele and they don't produce the alpha-actinin protein in the muscles. So research shows that having the alpha-actinin 3 protein in your muscle can protect you in the way from injuries and falls. So yeah, so there are also other genes associated with this, but alpha-actinin 3 has been one of those genes.
Okay, noted. And there is another more question from Dr. Yuli Dwi Haryani regarding genetic variation in SNPs. She asked about how do genetic variation in SNP contribute to differences in muscle power production among individuals, and how can the finding of this research presented be applied in the development of gene therapy to enhance fitness and athletic performance? So it's related to the genetic modifying because it's SNPs, yeah, and there's a changes in the base of the nucleotide. So additionally, is it possible in the future to perform genetic manipulation to achieve the desired level of optimal fitness and performance? And if yes, what are the ethical implications? It's a quite complex question, Prof.
Okay.
Yeah. So maybe you can share your thoughts.
Yeah, actually, the first step is like to identify those SNPs that are influencing certain fitness traits, such as muscle power or muscle endurance. So then to genetically modify humans. For health-related reasons, is in the future might be possible, but to genetically modify humans for increased athletic performance, that there are ethical reasons about gene doping, basically. So if you, so there would be gene doping is it's very difficult to be detected. So you have to actually know somebody's genetic composition, like the initial genetic profile, and then after he using gene doping to change. So that's a like this is considered to be unethical. So probably WADA has to revise its regulations about maybe not just collecting urine from the athletes, but also maybe have a biological passport of the athletes, which they will have their certain genetic polymorphisms in the very initial stages of their career. And then if something changes, then they should use also blood samples to track any changes in the DNA. But for health reasons, for example, with ACTN3 gene, there have been studies that use to genetically modify like genetically modified to produce the protein. The problem was like alpha-actinin protein was overproduced in large amounts. It was toxic for the muscle. So having some protein helps you producing more power. But if we increase the amounts of protein significantly, then it's toxic for the muscle. So it's not going to improve performance, but will destroy the muscle.
Okay, so it's kind of like there is a long-term effect of the SNP itself. Is it true, Prof?
If we modify the alpha-actinin protein in large amounts, then there is it's toxic for the muscle. So, but if somebody is XX, for example, and wants to become R and produce some amounts of protein, it's okay. So it has to be within the physiological limits, because the study I'm aware of, they try to increase the amounts of alpha-actinin significantly, so more than an RR individual would produce in his muscle. So this wasn't successful because it was toxic for the muscle.
Okay, interesting point. I also want to pose you a question based on your presentation regarding ACTN gene, ACTN3 genes, because it's related to the metabolic enzyme production. So I'm wondering, what would the SNP in this gene would affect metabolic activity of the muscle itself? That is the first question. And would it also increase the risk? Sorry. Would it also increase the risk of metabolic disorder due to the increased metabolic enzyme production?
Okay. So there are some studies that study the influence of alpha-actinin 3 protein on glycolytic enzymes. So yeah, and it has found that interacts with certain glycolytic enzymes. So probably the way that influences sprinting performance is through interaction with certain glycolytic enzymes.
Mhm. So is there any chance for the risk of metabolic disorder arise from this SNP?
Studies with
the ACTN3
yeah,
gene.
actually, this SNP can influence the prognosis of probably of certain metabolic disorders, and can also be involved or on prognosis of certain disease, any disease, so in the body, but can be involved as a contributing factor on a disease prognosis. For example, let's say in elderly, some people suffer from sarcopenia. So, but being RR maybe protects you and make you survive longer. Okay, increase survivance or also in metabolic disorders that could probably have at some point some influence, but is not the cause of the disease, but can influence the prognosis of the disease.
Yeah. So there is an additional factor beyond this that might influence the outcome of the athletes, yeah, Prof?
Yes, it should be additional factors, but this may be contributing factor.
Okay. Next to the further questions from Dr. Hendy Hafiz, we still have time. How do SNPs associated with muscle performance influence energy metabolism regulations in individuals with obesity at the molecular level? He wants to know about the molecular mechanism linked to the SNPs.
Of any particular SNP or
linking SNPs associated with muscle performance.
Okay.
In relation to energy metabolism regulation. Because the individual has obesity, which maybe has a problem as well with the metabolism.
Okay. There are certain SNPs identified in the leptin gene that can influence obesity. Okay. So leptin gene, leptin is a factor that has been well investigated about how this factor influence obesity. So there are certain SNPs within leptin gene that influence how someone can lose weight or gain weight. So I personally haven't worked with obesity and obese population, but there is a lot of interest on this area. So the SNPs related to muscle power and strength and endurance performance can have maybe a limited influence on how fast someone can lose weight. Okay, so the major influence and major impact would have probably leptin related genetic factors.
Okay. Maybe Miss Miftah, you want to add some response?
Yes. I have read one paper that correlate with the obesity. It is the polymorphisms in PPARG. Yes, this is the one of the peroxisome proliferator that will activate the I can say pro allele to linking to the higher BMI or metabolic tissue. It will impact for the obesity.
Okay. There is another more question. We just proceed because we have several questions to discuss. This is question from Prof. Ilmiawati. Is there any information in the difference in certain SNPs in different race? It's like in Caucasian, African, Asian athletes in certain sports, because racial difference seems to have advantage in certain sports.
Actually, there are differences between certain populations, like Africans, Asians, and Caucasians in genetics. So with regards to, let's say, ACTN3 gene, while there is some variation, there is variation in Caucasians and Asians, there is not much variation in Africans. So in Africans, studies with African populations, they found that there are all RR. There is no XX in Africa. So whole Africans, while in Caucasians, there is 20% XX and about 30%, 20% RR, and the rest is the heterozygotes. So in Africans, there are all RR. So it means that all Africans, apart from 0.5%, that is XX, so it's so rare. So these studies tried to be conducted there. They were inconclusive, so they couldn't do any comparison, because even non-athletes or elite athletes, Africans, most of them are RR. So that means that, I mean, while there are all of them, athletes or non-athletes, that this gene, there are other genes that make them being so successful, that we have to find them out. So, but it's not the ACTN3. Okay, but in Caucasians and Asians, the ACTN3 is as far as we know, contributes, but not in Africans. So this can also, I mean, depending how you look at it, it might mean that Africans are so good because they have ACT, all of them, they have actinin-3 in their muscles, but it's not actinin-3 that makes them so special in sport.
Okay, I got your point. So different race, they may have different, in different genes, that would also contributing to the athlete performance.
Yeah, that's an interesting, actually, observation, that different races have different genes that make them special. So the new trend in this field is like focusing on a race-specific, like research. So not in just general that these genes make you being a good athlete, but is race-specific genes that makes you special.
Yeah, it will be interesting for future research, I believe, Prof.
Yes, definitely. That's why working with Asian population is also interesting, because they have been less investigated within this field compared to Caucasians that have been heavily studied. So that's why I'm happy being in Asia.
Yeah, physiology department, maybe we can involve later for the next collaboration with Prof. Yeah, inshallah. And I saw chat in the box chat, Dr. Muhammad Reza, he wants to speak with Professor Ionis. Maybe is it through the Zoom or outside the Zoom, Dr. Reza? Okay, maybe we can do it outside of the Zoom.
Okay.
You can, yeah. We have the email of Prof, so that you can chat directly to Prof via email.
Okay. Great.
Okay, I think the session of the QA is finished. For those, for the remaining questions which cannot be covered for this session, maybe you can, or maybe we can help posing the question to the email of Prof or Miss Miftah.
Yeah, you can contact me by email about
Yeah.
anything, any question you have is okay.
Yeah.
So
As a final question, just one more. If a master student is interested in pursuing a doctoral program, what recommendation and requirements are necessary for admission to Mahidol University, particularly to those specializing in physiology? So this may be from the audience who are interested to continue for further higher education. Is there any potential collaboration opportunities available for international students in this field?
I think that question maybe
Is
Miss
it depend on the MOU?
can answer, because she went through this process.
Okay.
So she knows more about the detailed process. So what you should do to get accepted for as a student, but yeah. So
Yes.
can you?
Yeah, I just want to share my experience how to study here.
Yes. Please.
Okay. From the Master Biomedical Science, you can continue to come to Mahidol University, especially for the Faculty of Science. And for me, especially, I study in molecular medicines, but my laboratory is under Mr. Yanis laboratory in physiology department. So, in molecular medicine, it's also under the Faculty of Science and under the Ramati Body Hospital. It is the like collaboration of this faculty. So, but I got the scholarship from the Faculty of Science at the beginning. So, for you all that might be want to continue study here, for the future, you can start to do the future research for research plan, and and you can register by the website. All of the scholarships are available on the websites. Especially for the first year, you can apply for the young scientist scholarships, and for the second year, you can doing teaching assistant or some scholarship that you can apply here. And for the research, you can do the scholarship from the advisor and from the faculty science, and also from the Mahidol. You can apply for the scholarship, especially for the research. It's amount around three three 300,000 baht, for the amount of the research. But for the living allowance, you can also receive in every month for the living allowance here by the young scientists, and young scientists can cover all of the things of the course that you take here. So it is the chance that you come and that you can choose from the Mahidol scholarship here. There's also one scholarship that available now. It is the ASEAN scholarships, but I can for me I can register because I already in last year. So at the beginning, you can now apply for the ASEAN scholarship also in Mahidol.
So I personally accepting students. So if someone was interested in joining like this, we have so many genes to investigate
more.
Yeah. So if someone was is interested in this field, can contact me by email, and I can try to help her or help him to like find his way to here.
Okay. Thank you very much, Prof. And Ms. Miftahul for the information. That concludes our Q&A session. Thank you again, Prof. Yanis and Ms. Miftah, for addressing our questions so thoroughly. Your insights have been incredibly valuable and inspiring. We are truly appreciating the time and expertise you have shared with us today. So, and I would like also thanks to our participant for the engagement and thoughtful question. We hope this session has provided you with new perspective and knowledge to apply in your studies and research. So for those students who are now in the master degree, you can prepare your proposal and then you can contact Prof. Yanis just to see if it's matched with his project, and then you can join with him. Before I wrap up this session, I just want to take a moment to say a huge thank you for Prof. Yanis and Ms. Miftah. It's been an incredible, valuable discussion, and as a small token of our appreciation, we'd like to present this certificate of appreciation to Assistant Professor Yanis Papadimitriou as a speaker, and also for Ms. Miftah as a second speaker. Thank you again for being with us today.
Thank you for inviting me.
Thank you very much. We truly appreciate your time, and we hope have you join us again in the future. Big applause for the two speakers. Okay, now at this point, I will now hand the session back to our MC Yvonne to proceed with the next part of our program. Yvonne, over to you.
Okay. Thank you very much, Dr. Dewi Rusnita, for leading today's session so smoothly and insightfully. Now I would like to present a certificate of appreciation to our wonderful moderator too for her invaluable contribution. We kindly invite Dr. Dewi Rusnita and present the certificate as moderator of Guest Lecture 2025. Okay. Okay, ladies and gentlemen, we have explored valuable insight on single nucleotide polymorphism and its crucial role in shaping athletic performance. We hope this session has provided you with a deeper understanding and sparked new ideas for future exploration in this exciting field. Well, ladies and gentlemen, special thanks to our esteemed speaker for the time and expertise, and our moderator for guiding us through today's discussion. And let's give them a big round of applause. Okay, and I would like to thank you all for your time and active participation. We also had some lively and interesting discussion today and talk about opportunity too. Thank you for your active involvement contribution, and we kindly ask all participants to complete the post-test. It will be help us to evaluate how much you have learned and understood this lecture. Please access the post-test through the following link, and please take a few minutes to fill it out. While you fill in the post-test, I hope you have learned something new and useful from this guest lecture. Please fill out the feedback form too that will be distributed via chat room, and your feedback is important for us and the speaker too to improve our future events. I just want to remind everyone to stay connected with us on social media too. Make sure to follow us on Instagram at S2 Biomedic FK UNAND in Instagram. And thank you for you all for being here today. And certificate for the participants will be sent by the committee to your respective email address on the couple next day. So we look forward to seeing you again to future sessions. Stay curious and inspired. Have a great day. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.